Hello students, now we are going to discuss about the assessment of microbial contaminations and spoilage, right? So whenever any kinds of microbial contamination taking place into the pharmaceutical products or into the pharmaceutical aids, so we need to assess them whether it is contaminated or not in scientific way, right? So what may be the assessment generally in our home also, something if it will get spoiled, like your any food items or like any curry or any recipe, if it is getting spoiled or it got spoiled, how we are assessing? First we are seeing by the organoleptic evaluation, like color, order, taste, shape and size, or any changes if it is occur, we are trying to smell it, and then we are guessing that it has already spoiled, now it is not in the condition to eat or further use in that way. So there are in pharmaceutical, when it comes to the pharmaceutical formulation, we need to have some taste or some types of scientific assessment techniques that we are going to discuss. So here I have divided into the four category to assess the particular spoilage and contamination. The first one, it is the physical and chemical changes, right? So that's what I told you, any kinds of changes if it will occur into the state or into the form of the particular formulation. Say for example, change in viscosity, right? Say for example, honey or any syrup, it will be having its own flow property, or viscosity it will be specific. So we are checking that viscosity, if the viscosity is changed, if it is more or if it is less, then the particular range definitely it is spoiled. Then particular pH, what is the pH range supposed to be there with the particular formulation, if it is there into the range or not. If it is not there, that means microbial attack has been already occurred and it is spoiled. Like emulsion stability, what is that, the specific stability it is supposed to have. So that is, these all changes it will indicate microbial spoilage. Coming to the second one, sterility test, right? So when we are testing, like it confirm, this testing it will confirm the product is free from microorganism, right? So these techniques we have already discussed, the membrane filtration and direct transfer, right? Whatever the pharmaceutical products, if it is there, that product we are putting into the membrane, and we are dividing into two categories. Then we are going to incubate that one, and we are seeing the microbial growth, if it is there or not. If it is microbial growth, if it is there, means the product it is spoiled. In that way, in the direct transfer, we are transferring directly into the, and then we are going for incubation. So in this way we are testing the sterility, whether the given sample of the drug or medicine or formulation it is having microbial cell or viable cell or not, by incubation. And by after incubation we are checking the turbidity or the colony counting and so on, right? The third one, non-sterile product assessment, right? The product which is non-sterile, sterilization has not been done for the product. So for that, how we are going to do the assessment? The detection of the pathogen and total viable count, right? So what is the number of this microbial cell present into the that, for that microbial limit test we are doing, right? Again we have to go for with the product we need to take as a sample, and that we are going to incubate. After incubation we are going to count the number of viable cell present into that, and what is the limit for the particular product, how much microorganism can be there. So that we are assessing by this method, right? Coming to the fourth and important one, estimation of the pathogens, right? So pathogen we need to estimate. So there are two tests, BP pyrogen test and LAL test, right? So what is the BP pyrogen test? Here we are injecting it to the laboratory rabbit, we are animal volunteer, we are taking this animal, the laboratory animal model, where we are injecting, and we are seeing the body temperature is monitored, whether temperature it is getting increased, because whenever the pathogen or pyrogen it will be attacked to the body, the body will increase the temperature, right? The fever will come. So that we are monitoring, if anything it is there occurring, the body temperature if it is rising, means the into your injection or into your sample which you have taken from the pharmaceutical products, that is having microorganism. That's what it is. That's what this is. The next one, LAL test. That's very, very important. The full form of LAL also it may be asked, the Limulus amoebocytes lyosate, right? So this is the one of the what you call aqueous extract of blood cell of the Atlantic horseshoe crab, right? From there it will be taken. So what is the role of this lal? It will be react with the bacteria endocrine, right? In that it will and it will indicate the gram negative bacteria, whether it is there or not. So in these methods we will be confirming that the microorganism, if it is there into the pharmaceutical product or not, or the product has been spoiled or not, right? So this is the detection or assessment of the microorganism of the pharmaceutical products. Thank you.